take a minute to set up your page so you can take notes. You're again listening to Period 7's notes. They're kind of sleepy today. It's their Monday. Say hello, 7th period.
Hello.
Okay, so this is 119. We're in Unit 8, which is cells and viruses. And we're doing B4B, and we're just completing this. So if we look at B4B, it says that you're going to investigate and explain cellular processes, including homeostasis and transport of molecules. We've already done homeostasis, so we're going to focus on the transport of molecules. So investigate, so B4B, investigate. This is what our lab will be over tomorrow in class. So investigate and explain transport of molecules. My color didn't show up at all, did it? There we go. Okay, so when we do these teks, some of these teks they seem short, but they're really complex when you start to go into them, so I want to keep them separate and not just overwhelm people with some stuff here. So this whole thing is going to be just about transportation of molecules. And what we mean by that is just moving things in and out of the cell. It would be nice if we could say, hey, only the good stuff gets in and out of the cell and the stuff we want, but pretty much if it's chemically similar, then they can usually go through the cell. If they're chemically dissimilar, then they usually can't go through it. So when I say transport, we transport through a semi-permeable membrane. Okay, and I'm going to underline that. Do you guys underline stuff or put stars around it or circles around it when I do up here? Sometimes I'll be writing normally and then I'll write in big capital letters. That's really important too, because those things stand out when you go back and study for this. And when I say study for this, I mean you. You take the pages out, you go over these and you study these. When we talk about a semi-permeable membrane, we're talking about the cell membrane. Semi-permeable means it lets some things in and keeps others out. So again, it would be nice if we could keep all the poisonous stuff out and just take the good stuff in, but things that are chemically similar, like sugars and things. So for example, antifreeze. Antifreeze, you guys know antifreeze is highly poisonous, right? But it's got a sweet taste to it. It's a sugar base. And so if you left it, if you were really a terrible human being, if you left some outside, animals would come and drink it, and that would kill them. So if you were to drink it, it would get inside your cells and it would kill you too. Antifreeze would go in and out of your cell without a problem. So it's not just the good stuff that can go in and out of the cell, it's things that are chemically similar can go in and out of the cell. Things that are not similar to that can't go in and out of the cell. Ethanol, we talk about alcohol, people drinking. When they're drinking alcohol, obviously that's a poison. If you put enough in your body, it makes your nervous system wonky. You keep putting enough in there and it'll kill you too. In the same sense that rubbing alcohol like you put on sores or cuts or something, same thing. It'll just kill you a lot faster than the drinking alcohol, the ethanol will. So rubbing alcohol would be like propanol. It's got 5 carbons in it. Hang on. It's got 3 carbons in it. And then methanol would have, no, it's ethanol, would have, I think 2 carbons in it. Anyway, okay, so they're chemically similar. So they're both toxic to humans. but it's just one's more toxic quicker than the other. Yeah? So somebody said, hey, if I only put a little bit of the poison in my body, it'll just make my nervous system weird and I'll be drunk. Yes, that's what's going on. The other ones, they're so toxic that even if you put a little bit in your body, it'll kill you. So, yeah, I don't know who figured that out first, but there it is. So it lets some things in and out, but the alcohols, even though most of them are highly poisonous, they can get in and out of your cell easily and they kill you that way. So semi-permeable means some things can get in and some things can't, but generally you think if it's chemically like that, it can get inside the cell. Okay, so when we talk about the transport of molecules, I'm going to talk about two types of transportation: passive transport and active transport. Okay, so grab a different color. If you're not using colors, I don't know why you're not. It's really helpful in these things. I see most of you are still using pencil, that's fine, but you need to designate somehow. If you can write differently, like here, have you ever seen a paper and it's got different fonts and different paragraphs? It's really handy to help you remember things like that. Sometimes you can write forward like this, or sometimes you could print and in the next paragraph you could do cursive. Things that make them separate from each other, colors make that really separate from each other. So, two types of transporting things in and out of the cell. And the first one is passive transport. Passive transport, no energy is required. That's the key there, no energy required. This is natural diffusion. All of these things are natural, but when I say natural, sometimes I just think it just happens. Natural diffusion and osmosis. This occurs or goes with the concentration gradient. Okay, so I'm going to go back one. I don't remember, it was Friday, it was a long time ago. So concentration gradient. Right here in the middle of the page, right here, concentration gradient. This is when you move from a greater concentration to a lesser concentration. Remember those square brackets mean concentration, that's how you read them. So they move from a greater concentration of the chemical to a lesser concentration of the chemical. It's kind of like going downhill, or it's kind of like you throw something in the river and it floats down with the river, right? It requires no energy. You throw it in the river, it's going to... Have any of you guys ever gone canoeing or floating on a river? Yeah. And you go with, if you try and go backwards against the flow of the river, it requires a lot of energy, right? That would be active transport. You have to supply energy to it. Passive transport, you throw it in, it's just going with the river. Okay? Now, in this case, passive transport is what happens when you don't have to supply any energy. It's just naturally, on its own, going to happen, requiring no energy whatsoever. Now, there is a special type of passive transport called facilitated diffusion. Big ugly words. Facilitated diffusion. And facilitated diffusion is the diffusion of molecules or ions through the proteins in a cell membrane. I could just put membrane, I ran out of space there. Do you remember what the cell membrane looks like? It's a double layer of lipids with big chunks of protein in there. Okay, so up here at the top, I want you to put page 17 in your packet because I want you to make sure that you turn to that when you need to. It's got great pictures in it. So I'm flipping over to page 17 in your packet. And for you guys online, it's the Star Tune-Up B4B cellular processes. So this has the pictures in it. So when I look here, this is a beautiful picture of the cell membrane. So here we have a double layer of lipids. So look, there's a layer of lipids, it's the balloon with two strings on it, and there's the second layer of lipids upside down from that. So we have two layers of lipids, and here is a protein chunk, and there's a protein chunk. They don't have to be the same proteins. Different proteins are in there. So we have protein chunks that are in there. And so most things, if they can get into the cell, they'll just diffuse through the lipids, in and out through the lipids. But some things, they can't get through there. They won't dissolve in that stuff. They can't get through there at all. So what they have to do is they have to go through a special door. And it limits how much stuff is getting in because there's just a limited number of doors. Hey, wake yourself up. If you need to stand up, do so. But you got to focus while you're in here. So let's say, for example, I have a big football field and I have no fence around it. Just everybody's going to get in anyway. Okay? So everybody can get onto the field all over the place, right? Pretty quickly, pretty fast. If I have the same football field but I put a fence around it and only two gates, one at the end at each end, how fast are people going to be able to get in? It's going to happen a lot slower, right? They can still get in, but it's going to happen a lot slower here. So think of those proteins as kind of like gates into the cell, so you have to go through a special gate because they can't go through the lipid bilayer. They have to go through the special gates to get in. That's what facilitated diffusion is. They're still just diffusing in, going from an area of greater concentration to an area of lesser concentration. It's still diffusion. It doesn't require energy. It's just that they can't get through this part, so they have to get through one of the doors. Yes? Yes. Okay. So page 17 has some really good pictures. I'm going to refer to this again. You guys on remote, you might want to grab a picture of that on your iPad or on your phone so you can refer to that too. It makes it much simpler to do that. The next part we're going to talk about, pick another color. Let's see, where's my other stuff? Oh, they're there. Let's go orange. Okay, the next part, we've done passive transport. Now we can look at active transport. If passive transport, there's no energy required, what do you think active transport is?
Energy required.
That's exactly what it is. This requires energy. So this would be like trying to go upstream on the river. You went out on the river instead of just floating down the river with the current, that would be passive transport. What you're trying to do is trying to go back against the current. So you're paddling against the current back. Requires a lot of energy. Okay, so when we have active transport, it's going to require energy. And what is that high energy of the molecule of the body called? What molecule is that called? ATP, in the form of ATP. ATP. Now, when we have active transport, this is the movement of the molecules against the concentration gradient. You notice how I keep using these phrases over and over again? Those are phrases that you're going to see. So when you see all this, when you read these questions I pop here on the top, you'll notice it's got a lot of scientific terminology that they use. You can't let that throw you off. You have to take that into account. You have to understand what those things mean. So this is the movement of molecules against the concentration gradient. Gradient, I can't spell it now. Gradient, there it is. There we go. So it's against the concentration gradient. Here's the word right here that's important. So there are two ways of describing this. we talk about active transport, you can talk about the fact that it requires energy, and 2, you can talk about the fact that it's moving molecules against the concentration gradient. So 2 ways to refer to that. And there are 3 that are going to be important for you to know. The first one is called the sodium-potassium ion pump. So far I've been really disappointed at the number of people who don't know their symbols for the elements. Anybody know the symbol for sodium?
Na.
Na, thank you. What substance do you know that has Na in it?
Salt.
Salt, sodium chloride, yes? Symbol for potassium?
K.
K? Alright, so let me tell you this. If you don't know those 2 symbols, you're going to die next year in chemistry. I strongly suggest that you get a little app on your phone. It's a periodic table. It's free. And you can go through and quiz it on the symbols. You better know them by the time you get up there, because the symbols, that's the easy stuff. You're going to have to name compounds, write formulas, balance equations. Balance equations is easy. You're going to have to write I'm going to give you a formula, or not me anymore, but they'll give you a formula and say, what is this? So you have to know how to name these things, and you use that by looking at the symbols and figuring out what the names are. If you have to look up every single time and figure out, well, what is that symbol? What is that symbol? It's going to take you forever. You're never going to get this. Get yourself a little app on your phone and just quiz yourself on the symbols for the elements. If you don't, I don't want to hear you whining about it next year. Okay, so we have active transport. The first type is the sodium-potassium ion pump. And this is actually kind of like when the protein in there acts like a little machine. So the protein that's in the phospholipid bilayer, right? The protein, and I'm going to put machine in parentheses. I always think of the molecules as these little machines that are working, because they're big molecules and they have these oddball shapes, and their shape determines what they do, and they change shapes and move almost. So they're really pretty cool. The protein in the cell membrane uses ATP to maintain the sodium and potassium ion against the gradient. Okay, sodium, ion, and remember when I put square brackets, how do you read that? Concentration. And potassium, ion, and ion is an atom that has a charge. You know that electrons equals protons usually in a neutral atom, right? Yeah. When you have an ion, you've popped off an electron, or you've added an electron to it. When you have the potassium ions, they're not by themselves. They are in a chemical compound with something. Like sodium chloride, NaCl, salt, that you put on your fries. If you put that in water, it's easiest to see, but when you put it in water, they separate a little bit from themselves, and the sodium gives up an electron, the chlorine kind of takes it, so the chlorine side is a little bit negative and the sodium side is a little positive, and that's why you can run electricity through salt water. Yeah, they have ions, electrons in there. So we have this protein that has to maintain the sodium concentration and the potassium concentration against the concentration gradient. So when I look at page 17 here, on this picture, do you see how all the sodium ions are over here and the potassium ions are over here? If you look closely at your paper, you can see this. So as your cells are sitting there right now, that's the way it really is. Normally, they would be equal on both sides, right? Because they diffuse, so their concentrations are equal. But you're actively putting energy into the fact of keeping the sodium ions on one side and the potassium ions on the other side. At some point, for example, if you touch something, the way that the message from your finger, the neuron in your finger, goes all the way up to your brain is because these ions swap. Now, there are some negative ions associated with that too, but basically what it does is it changes the electric potential, the voltage on your cells, and what it does is it runs all the way up. It happens in less than a second. So when I touch something, it's almost simultaneous that I know I'm touching it and I touch it, right? Yeah, when I touch something, that triggers, that message has been sent. So all of these flipped in that less than a second of time I felt that touch, which is amazing. What that means basically, and it's all about the ions, it means that your body runs on electricity. So if I got a battery, and there's some wires up to it, and it's like in here, I can get a PhD. One of the most interesting labs I did in college, yeah, the frog's gonna die. So we took this frog and we pithed it to the PITH. And so you take a flat scalpel, you stick it in between its vertebrae, and you basically cut the spinal cord until the frog's not feeling any pain, but it's still alive. And then if you take the skin off its leg, there's no fat tissue here, so it's just straight muscle. And you get a battery and some wires, and you can make his whole leg jumping. Runs on electricity. Doesn't even have to think about it. It runs on electricity. Can I do the same thing to you, or people would not be happy with this? Yeah. You run on electricity. Have you ever heard of people getting shocked? They grab a wire that's hot and their hand clutches on it. Electricity is what you do. Electricity is doing that. You've heard of people. What is it? They're trying to recover from maybe a knee surgery or something, and they put those little electrodes with the battery pack on there, and all it's doing is sending a little tiny pulse through there, and it causes your muscles to contract and relax like that. It's strengthening the muscle doing that and making it work here. We run on electricity. So our nervous system runs on electricity, our muscular system, all of our muscles run on electricity. So we could, if we mess up our electrical circuitry, then we're really screwed up. Yes, yes, that's exactly why. Okay, so we have this active transport that requires energy. So we're going to move stuff against the concentration gradient, and we use it heavily in the body here. Now, besides the sodium-potassium ion pump, which is really important, you have two other things, and I'm not sure if you've heard of these. Have you heard of endocytosis and exocytosis? Okay, so endocytosis. Okay, we've talked about pieces of this. The prefix endo means in. Do you remember cyto? Anybody? Cytoplasm, cytology. Cytoplasm is the what? What is cytoplasm? Yeah, it's the cell jello inside the cell. So cyto means cells. So this, if we translate it, it means into cell. So it's going into the cell. When you talk about endocytosis, it's a way of getting something into the cell by reaching out, grabbing it, and engulfing it. And kind of like they reach out and bite it down on it and engulf it in there. It's kind of sort of like that. This is when material from the outside is engulfed. And taken into the cell. And let me show you a picture before you write the rest of this down. So look at this. Here we have, let's say we'd have a straight cell membrane, just kind of going straight across there. And there's some chemical that touches it. Maybe it's too big, maybe it's something weird, but the cell decides it needs it. And so what happens is there's a little pouch that forms, like it dents in, and so that molecule kind of goes in that pouch, gets into the pouch, and the pouch pinches off. So it's like a big pinch off of the cell membrane. And this thing right here is a vacuole. That's how vacuoles form. They're little sections of that. So now what you have is you've taken this into the cell within a vacuole. Again, page 17, look at that. Okay, so white blood cells, you know you have lots of different types of white blood cells, right? So white blood cells, one of the most common is the macrophage. Macro means big, right? Opposite of micro. And phage means to eat. So it says white blood cells, and what they do is they crawl around. They crawl wherever they want. So you have these cells, your own cells that can crawl around wherever they want in your body. And what they do is when they bump into bacteria, they eat them. So exactly what I showed you here, the bacteria are really little, maybe there might be a clump of them, but a little section indents, and it's almost like it opens its mouth and swallows it up and takes it in. And then they digest it. So that's how they kill the bacteria, they digest it like that. Once they kill enough, it actually ends up killing them. Once they have so many in them that they're digesting them, then it kills those white blood cells. So when you have a big infection, some of that, when you have pus that's associated with that, you're going to have dead bacteria, but you're going to have a lot of dead white blood cells that are in that pus also. So that's what it does on there. But they do that. They kill these bacteria using this process of endocytosis. They are basically engulfing them and then digesting them. A lot of single-celled organisms, this is how they eat their food. They're only made of one cell. It's not like they have a digestive system. They have one cell, and so they're whatever they're going to eat butts up against them, and they engulf it and then they digest it in there. Same way as your white blood cells would do. Okay, the opposite of endocytosis would be exocytosis. Exo means out, so I'm going to put out of the cell. So that's what we could translate it to. So exocytosis, out of the cell. Same process, only backwards. Okay? In this case, you have a vacuole with stuff in it. Maybe it's waste products. A lot of times, though, it's stuff that your cell's making on purpose. It's not just junky stuff. It's maybe proteins that need to get out. But you have this vacuole with stuff in it that merges with the cell membrane. And puts stuff outside of the cell. So again, page 17. Notice I'm looking at page 17 again. Here we have endocytosis. And look at this one. Here we've got, and I don't know why they didn't put their stuff right in here. Here we have endocytosis. This over here on this side is exo. So we have something in the vacuole. It's moving up. It actually butts up against the cell membrane, and the cell membrane starts to open, and then it just pushes it out. And so it ultimately moves back out. So it's just the opposite of what you had here. Yes? So it's a way of getting it out of there. It's not going to digest the stuff that's here. A lot of times they're making proteins and they want to send them out of the cell so they can go to other places. The bloodstream carries it around, so it does that. So we have endocytosis and we have exocytosis. All right, so 3 examples of active transport: the sodium-potassium ion pump, endocytosis, and exocytosis. Okay. Now, I want to give you a couple of other examples. How much time do I have left? Oh, okay. A couple of the other examples. There are 2 main examples that I want to give you that you might see on the STAR test here. The villi of the intestines. They're the. You guys know what shag carpet is? Yeah. Okay, so it's like you have these microscopic little shag pieces of carpet that line your small intestine. And so you digest your food mainly in the small intestine, and then all that food is absorbed through these little pieces of the shag carpet. They're individual cells, and they're absorbed in there, and they're lined with blood vessels. And so this is where you're taken in. Here's the reason it's active transport, okay? So once you start eating and digesting, you're putting a lot of the sugar and stuff, the nutrients, into the bloodstream. Well, midway through, when you've digested midway through, your bloodstream is loaded with that stuff. And it doesn't normally just keep diffusing because it would be going against the concentration gradient, right? Your bloodstream's got as much as it's going to hold, and you want to put more in there. It doesn't work that way. So what you have to do is you have to use energy and force the food nutrients from your small intestine into your bloodstream. So it keeps taking it around to all the cells like that. So this is going to use active transport to absorb more nutrients. Another example would be root hairs on plants. Okay, so you know the roots are going to absorb the water, right? So the root hairs on plants, they will absorb excess water. You already have plenty of water inside there, and they're going to take in even more water. So they'll absorb excess water into the plant against the concentration gradient. That's active transport. When you go against the concentration gradient, you have to use energy That's active transport. We have about what? Okay. Okay. Okay, once you have that down, go ahead and pack it up. There's some weird stuff. I mean, those white blood cells, they just crawl around everywhere. That's crazy. Yeah. It always makes me think somebody's going to come up with a different kind of endosymbiotic theory. Like, where did those white blood cells come from? That can crawl around on their own. That's interesting. That would be some massive bacteria.
Hey, we haven't discovered all the bacteria.
Nope, we haven't.
We don't know what's going on. And the world was at one time mostly ocean, and we haven't discovered most of the ocean.
A lot we're missing. Yep. Okay, I don't know if I'm going to like this whole setup because you guys keep moving up here. Everybody pull back some, please.
Me, Berkeley, and Bailey won't be here Wednesday or Thursday because of stock show.
Okay, fine. Your stuff will be online, so when we go over that stuff on the virus, yeah, you guys just need to be working on that,
And
okay?
the lab, will it be online also, or?
No, the lab's in class, and we probably won't have stuff left over, so I'll exhibit you guys with that. Just 3 of you guys? Yeah, okay. Thank you, you too. Daniela, if you're up closer, you have to talk more. Yeah? Yeah.
No, she had basketball too. She's like a manager.
Alexia's basketball. Miriam's here. What about Eliza?
I saw her at 6th hour.
Maria?
Oh, she might have went to the orthodontist.
Yeah.
Cause she told me. Oh yeah, cause she told me something about her braces.
I don't know. She was in 6th hour, but she left early.
Right, Ramsey's in here. Wait, oh, I forgot that she was in this class. I always don't even know who she was in.
There's like 8 of us in here.
Gosh. Oh, my classes have been small today, except for my 3rd hour.
Let's see. I have several of you guys. Papers in the paper. One is page 17, 13, 14, 12. And the other is a draft lab paper from last week. So,
Oh, shoot.
that'll
Get my desk. I think I'll turn that one in. The loopy one. Wait, didn't we do the loopy one like as like a group or something like that?
as a lab paper, you did your own.
Oh, so then I think I turned.
Thou shalt not.
I think everybody told me.
Yeah, yeah. But I think I turned mine in though.
Miss Howard, you spelled my name wrong
Where?
on my sticky note.
Enersyn. Is that not
Put
right?
an O.
Oh, okay, sorry.
It's okay. Everybody does it.
It's
On my
okay.
baby blanket, I have this baby blanket and it says ENNERSON. There's not even two.
You should see how the school spelled my name on my lunch card. They butchered it. It was terrible. It's like MC capital K ENIZE or some weird stuff like that. I don't even know. I still haven't fixed it since like 8th grade.
Okay. Let's see.
Yeah, I didn't turn this in.
Hey, that's not. Hey, you have
finishing the lab. Now your lab paper has a whole bunch of stuff to it. I don't know that we'll have time to go through all of it, but we'll do the lab part and we'll get through as much as we can. But hopefully on Thursday we're also going to do another part of the notes on there. Oh crap! I forgot to stop my note taking. Oops.
Oh, we're supposed to finish our notes today.
Thank you.